Top Bed Talk.
I should reintroduce myself, Monty Mython, the founding director of evidence-based perioperative medicine, going into our 25th year. How good is it to be here? So great to be here. Welcome to UCL, which is a sort of home originally of EBPOM, but the first time we've actually held the meeting here, in part because it's the 25th anniversary, but also because last year was the 175th anniversary of the first general anaesthetic given publicly in Europe here, or at least, for advertising, marketing purposes, it was right here. So without further ado, I'm going to introduce our first plenary lecture of the day. It's the Ernest Henry Starling Plenary Lecture. There's my friend Hugh popping up there. Morning, Hugh.
Morning, Monty.
And Hugh may well be talking about Starling in a moment, but Starling's laboratory is just along the road here. Obviously, Starling sadly left us early in the last century. But of all his great work, the forces, etc., what happens with the Frank Starling curve in the heart. Obviously, Starling's name was not Frank, it was Ernest Henry. Frank was the other guy. So, Hugh, thank you very much indeed for doing this morning. Hugh is very pleased to describe him as an old friend and a colleague. We actually went to medical school together. The first time I met Hugh, which is the nature of the man, he was submerged in a swimming pool in a scuba suit, breaking the world underwater playing record. And that was in the nurses' home swimming pool. So well done for that, Hugh. I don't know if it still stands in the Guinness Book of Records.
It does.
The other thing, Hugh is a professor of cardiovascular genetics and critical care here at University College London. We were actually at medical school together at the Middlesex Hospital, which is just over the road, founded by the same anatomist back in the days, so a few blocks away, but actually competing at the time medical schools, but now joined as one. Hugh has a long-standing, impressive research career, very impressive research career, dealing with, for example, things like the genetics of human fitness, so his original Nature publication in the last century, telling us about the ACE genotype and its relationship to how humans become fit or not fit in different ways. Human tolerance of hypoxia, for example, one of the founding leads of the Xtreme Everest team, of which we've got Dan Martin, Mike Grocott in the room at the moment, who both summited and took blood gases from the summit of Everest, but a lot of that research stimulated by Hugh. And also a passionate, many other subject areas, but passionate about climate change. He's also a passionate clinician practicing at the bedside in critical care, and was one of our national leaders for COVID-19, so often appearing on the media, on TV, radio, and in print, making some very challenging observations about the challenges we faced with COVID-19. He did that as a board member of the Intensive Care Society, so thank you for that, Hugh. And in recognition of all of that, the Queen recognised his contributions with an award, one of those letters we put after our name around here, that happens, British Empire stuff. So well done for that, Hugh. Want a further explanation of that later. We've got a few others in the room that can explain it. So without further ado, I'll hand over to Hugh. He made a massive contribution to evidence base. He's going to give you a personal view on that in a lecture I think he's entitled, I Know Nothing.
Thanks very much indeed, Monty. Indeed, I thought I would reflect on evidence base, because Monty said we've been trying to practice evidence-based medicine, and I've tried to make a contribution to that. But my contribution actually is to learn that over 502 months that I've now been in my medical career, I do indeed know nothing. This takes me back to the year that I started at medical school. Monty and I were of the same era, as you've heard. So on the left, this was the fashion of the time. And on the right, this is what your average rugged male looked like or wanted to look like. And I think I probably thought that that's what I looked like. And the reality was somewhat different because this turns out to be my medical school entry photograph at the time. But it's less what I wanted to look like then, but what I wanted to become. And I think as time went by, I wanted to perhaps not look like this character, but to be like him. I wanted to be a superb diagnostician, to be the guy who could nail the diagnosis that no one else could make. But the first problem I've learned is that the diagnosis we make is often, and I would say probably generally, not a real diagnosis. And if it is a real one, it's not correct. And we have some evidence for this. A study done in 2013 where they gave some web-based history examination and special test results to a bunch of interns and general physicians. And 2 of them were pretty easy, and 2 of them were considered a bit more challenging. And for the easy ones, 72% of the time, the respondents were certain that they were right. And for the hard ones, they were certain they were right 64% of the time. In reality, they were right half the time, or actually only about 6% of the time. And the problem with this is we all do this. We make a diagnosis, we then seek evidence to support the diagnosis, and if the patient does well with the treatment we instigate and recognise that the diagnosis was right, and if the patient dies, then it was a shame because they obviously died despite the fact we'd given them the best treatment, not because we'd actually given them the wrong treatment. And we don't recognise that in our own practice. That's true of me as well. We also know that we make mistakes in pretty much every other test we do. If we look at clinical radiological series, on average in most of the literature, up to 5% of the diagnoses are factually incorrect. We know that up to 25% of them in some series have errors in them. And we know that in up to 30% of breast cancer cases, they're missed on mammography. And that's why I can't be with you today. I'm with my sister with metastatic cancer from a missed diagnosis that was there in retrospect and was missed for a year. We know that in the emergency department, it's about 1 in 6 plain films are misdiagnosed. And we know that a third of CT scan reports have errors. We know the clinical pathology is often wrong, up to 25% of the time for lymphomas and sarcomas. And when we look at clinical diagnoses, when you get expert panels to review the diagnoses made, it can be up to 12% of the time. The diagnosis with which they're admitted is actually the wrong diagnosis as judged by expert peers. And as we'll come to, that doesn't necessarily mean that that diagnosis was right either. The ground truth, of course, is probably the only way you can really get to the bottom of it, and that's with autopsies, which we don't do anymore, do we? We don't get postmortems done anymore. But when you did have postmortems, you found that nearly half of cases had major discrepancies. A fifth of them would have changed your management. Half of them had discrepancies in minor diagnoses, of which a third were clinically relevant. And half of cases of TB hadn't been diagnosed. Over half of cases of pulmonary embolus hadn't been diagnosed. 35% of the aortic dissections hadn't been diagnosed, and half of the cases of cancer hadn't been diagnosed. So we don't have a ground truth. We miss these things, and because we don't get routine postmortems now, we're all living in a learning curve which is completely flat because there's no feedback. And all of this is because we're ignorant or stupid. There are things I don't know. It's because conditions can share symptoms and can lead us to make a diagnosis that's not correct. It's because of the confirmation bias of which I spoke. And it's because we get tired and fatigued and irritable about things that's going on and distracted, and we make mistakes. It's also because we're being asked to do things that we aren't designed to do. If you look at the panel on the left, they are identical twins, but every person in the room can look at those and realize they're two different people. You can instantly know they're not quite the same. And that's because your brain is tuned for recognizing people. They're also tuned for recognizing emotion. Is this person a potential mate? Are they sick that can make you ill? Are they aggressive? Are they a risk to you? We're meant to be good, women probably better than men, at reading emotional states from faces. That's what we're good at. We're not designed for anomaly detection. And unless we are specifically trained for that anomaly detection, even then we miss things. So this was a study done in 2013 where these CT scans were presented to expert thoracic radiologists, and they were asked to look at the scans, and the images were slightly doctored along the way, and they were asked to identify the major flaws. None of them, really, very few, picked up the gorilla that had been inserted into the images. And in fact, you may not be able to see that well projected at your end. It's always hard to know with x-rays, isn't it? But this is a bloody obvious great big gorilla. And it was missed by 8 out of 10 because people only see what they want to see. They only look for things that they think might be there. And often, as I say, with mammograms, because things are subtle, even when you know what you're looking for, you can get it wrong a third of the time. So the first issue is that even when there is a diagnosis we wish to make, it's often incorrectly made, and we never find out that it's incorrect. We just pat ourselves on the back that we made the right diagnosis. But it's much worse than that. We don't actually know what the diseases are that we're going to diagnose. And you may say, well, come on, Hugh, we absolutely do. And you'd be right for some things. Appendicitis in the world of perioperative medicine, you can be pretty sure, epigastric pain that moves down to the right iliac fossa, that's tender with guarding. Nowadays you get a CT, but of course the old days just open them up and there's a big red dangly thing with pus coming out of it. That's appendicitis. But of course that's not what most diseases are. Many diseases are actually syndromes. And if you look at the definition of a disease, it's actually defined as a group of symptoms which consistently occur together. And you say, well, that pattern of symptoms and or signs defines a particular disease state. But to discuss that disease state, we have to give it a name so that we all know what we're talking about. The trouble is, as our good friend of Mike and mine and Monty's, Lindsay Edwards, always says, these names were generally attributed to these diseases by Victorian gentlemen in top hats. They came at a time when medicine lacked the tools to actually define a disease in some biochemical, physiological, or anatomical way. So these labels got attached based upon nothing more than symptom clustering. And of course, many of the diseases are just names that got attached, and it's a con. You and I know this. We say to a patient, you have anorexia. Well, all we've done is attach a Latin name that they're not. Acne rosacea just means it's a red facial eruption. Rheumatoid arthritis comes from a pain which flows from joint to joint, which is just stating the bleeding obvious about what the patient's already told us. And asthma, when the patient says, I'm breathless and wheezy, what we say is you have asthma, which just means laboured breathing. So all we've done really is take the symptom the patient told us about, turn it into Latin, and attach that Latin name to the symptom they gave us in the first place. It doesn't actually, of course, define anything about a disease specifically. And if you go to Wikipedia, this font of all knowledge, this is just the list of the first part of the A's of these diagnoses. And you'll see that most of them are just syndromic, where someone, Grocott disease, Mython syndrome, has just stuck their name to a group of symptoms. It doesn't mean it's a disease. But of course, the drug efficacy, what we actually do, doesn't depend on the label we gave it. It depends on actually what the underlying pathogenesis actually is. So let's look at achy joints, a patient who comes to a doctor and says, I've got achy joints. So we take that, we turn it into Latin, and we give it back to them as a diagnosis. Arth being your joints, itis meaning inflammation. We say, you've got arthritis. And they say, wow, just as well I came to you, doctor, you're very clever. And then we do a rheumatoid factor. And if it's positive, we say, you have rheumatoid arthritis. And if it's negative, we say, you have something called seronegative arthritis, which is all right, except that, of course, a lot of seronegatives can become seropositive. So they probably had rheumatoid arthritis all along. Or if you just do it by the definition, you've got to have rheumatoid factor, they didn't have it, they had something else and then developed rheumatoid arthritis, which is clearly nonsense. If we then add in a butterfly rash, we go, well, that's very typical. And if we have got rheumatoid factor negative and we look for anti-double-stranded DNA, we say, you have something called SLE. And if you don't have SLE, unless, of course, you seroconvert at some point when suddenly you do. And remember that these sorts of tests were pretty rubbish. Rheumatoid factor is an antibody to IgG. It was discovered in 1940, before they even knew what an antibody was. It was 8 years before we even knew what they'd found was an antibody, because we didn't know what it was. And they only found it because out of all of the proteins in the blood, there weren't many techniques for looking for things. So it was a pretty mediocre test in the first place. And out of the billions of potential protein variants in the blood, they chose one, and we've ended up calling it a disease. But remember that those tests, the sensitivities and specificities and positive predictive values, differ, let's say for anti-double-stranded DNA, depending on what test you use to find it. So you can be no SLE with one test and SLE with a different test for exactly the same biological marker. And the same applies for things like anti-nuclear antibodies. So this is a mess. We've ended up giving labels to people and defining treatments based upon stuff that happened 70 years ago when we didn't really understand biology. Of course, with those, we then give people drugs. And we say, you've got rheumatoid, but you're a responder to your methotrexate, or you're a non-responder. Well, it might well be, of course, that that's not what was going on at all. There are now plenty of other tests, of course. So rheumatoid factor was 1940, but we've now got all of these other particular proteins and many more that can be biomarkers for rheumatoid arthritis or not. So which one are we now meant to use? Are we meant to use all of them? Are we meant to use some of them? And what do we use as our gold standard of diagnosis? Is it Monty Mython said it was, therefore it must be? Or is there some other way of defining the disease? And we're now into a circular argument of how we define the disease. Is it defined by gold standard Mython test, or is it defined by some of these other tests, which we're linking to a diagnosis we don't know how to make? We're chasing our tails. Rheumatoid factor, of course, turns out to be found in a bunch of other diseases we now know. So if you go looking, you can find rheumatoid factor in malaria, leishmaniasis, dermatomyositis, if you had a COVID vaccine, if you got infectious mononucleosis or Hep B. None of those are rheumatoid arthritis, and yet if you presented with achy joints and you did a rheumatoid test, that's the label you'd get, when in fact you might have hepatitis instead. So is SLE one disease? Is rheumatoid arthritis one disease? Do the different antibody profiles mean different diseases? Are non-responders and non-responders actually just people with totally different diseases? We don't know. This is an example Mike Grocott gave me, because Mike, apart from being an expert in pretty much everything, turns out to be an expert in the breeding cycles and evolution of gulls. And he told me some years ago about this classic example of a ring species. So basically, if you look with an American herring gull on the left of the picture to the west, it flies over a bit of water, one or two of them do, and they land on an outcrop. But the animals that landed there had maybe, let's say, slightly longer beaks. So they grew and they ended up being called a different sort of gull. And that went over a mountain range, and they may have had, let's say, bigger feet. So they had a little inbreeding effort, and each of these colonies ended up looking a bit different and were called a different thing. But the idea was, in fact, it was nothing more than different animals. It was just the fact you've got selective animals moving to different domains geographically and breeding there. So we've got this beautiful example of evolution through migration. And it turns out to be absolute nonsense. If you do the genetics and actually go for the biology rather than what you see, you find out this is not true at all. It's just nonsense. I would say the same is happening with us grouping things by we're saying, oh, well, that disease has a longer beak or bigger feet. It's only when you start drilling down into the biology that you really understand it. And here's a classic example. This is Lou Gehrig's disease, amyotrophic lateral sclerosis, motor neurone disease. Every single one of you in the audience would recognize that case. We all know what motor neurone disease looks like. We all could identify it. We all know what the signs and symptoms are. It's dead easy. Except it turns out that 10% of cases are inherited. 90% of them just turn up from somewhere. 10% of the sporadic ones have mutations that cause the familial version, but 90% of the sporadic cases don't have the genes at all that are in the familial one. So they're clearly, the sporadic and familial are generally not in any way the same disease. Furthermore, it turns out that there are at least 20 genes we know of that cause familial amyotrophic lateral sclerosis, and you can get the disease in any combination of two or more of those genes, which are spread across a vast array of different chromosomes. So they're totally different proteins, encoded in totally different parts of the genome, in totally different combinations, and we call that one disease because it ends up looking the same at the end. Furthermore, some amyotrophic lateral sclerosis is complicated by frontotemporal dementia. We say that's part of the disease spectrum. Well, is it? On the left-hand panel there, going upwards from the x-axis, you've got genes related to upper motor neuron symptoms in this single disease, we're calling it ALS. And you'll see there's a vast array of different genes that are associated with upper motor neuron features. If we go downwards, go south from that line, there's a whole bunch of totally different diseases that are associated with lower motor neuron features. If we then go across the x-axis, there's a whole vast array of different genes that are associated with the development of frontotemporal dementia. And you could choose any combination of 2 or more of those genes, and you would say that the patient has amyotrophic lateral sclerosis. Well, do they? I don't think they do. I think we're giving it a label of a syndrome, but not actually a disease. And if we look to the right again, we can see this clustering of these genes that also cross over into totally different diseases. So the big circle in the middle is ALS, but there are some with lower motor neuron symptoms we've talked about in clustering. There's a different disease state of hereditary spastic paraparesis. There's a bunch of mitochondrial diseases as well. These are what we would say are totally different diseases, but they share the same gene mutations, but they present in a different way because the gene combinations are different. Are these the same disease? As a consequence, we have been useless at finding a way to treat this disease called ALS because it doesn't exist. We've got one drug, and it's the only one licensed in the UK, and it's pretty mediocre. It impacts survival by about 2 to 3 months. And furthermore, we don't even know how it works. There's been a whole raft of mechanisms postulated for why it might work, but we don't know because this drug was not developed based upon a mechanism of disease. It was done by drug screening and chance. No surprises then, if we don't know what the disease is or what causes it, that we can't find any effective treatments. The same applies to a bunch of diseases that are much more common. So here's asthma and the genes that we know are associated with in the left-hand panel. At the top there are a bunch of genes associated with atopic dermatitis. On the left, a list associated with asthma. On the right, a list associated with allergic rhinitis. And the bottom, people who become allergic, let's say to house dust mite on their skin, for instance, or nettle rash. And you'll find that a lot of these genes are often associated, and you can therefore end up with an asthma gene and an atopic dermatitic one, or an allergic rhinitic one, or a sensitization one, or any combination of the above. So these diseases, is asthma one disease if you get it through a totally different gene? And if you get it with an atopic thing, is that the same disease? Because you've now got a combination of diseases which are interacting, or in a combination of disease-causing genes. I'm not sure that they are one disease. And furthermore, just as we saw for ALS, the genes overlap with totally different diseases. Some of those genes relate to depression and neuroticism. Some of them relate to hypertonia and indeed to increased blood vessel wall tension as well. Some of them are related to celiac disease, inflammatory bowel disease, and some of them are related to rheumatoid arthritis and diabetes. Again, same disease, different diseases. Is rheumatoid arthritis with wheeze asthma, rheumatoid arthritis, or is it neither of the above? Is it a different combination of genes that just happens to present with achy joints and wheeziness, and it's a totally different disease from any others? And indeed, a review in 2015 pointed out that asthma isn't a disease. They describe it as a collection of several distinct diseases, or endotypes, often presenting with varying phenotypic expression. Young, atopic, obese, middle-aged, or elderly, for instance. And we now know that. If we look at the phenotype, we know that the molecular mechanisms are very different. There's T2 high. Those often have high eosinophils, and they are mediated by very high T helper cell activity. But that falls into a group that's atopic, where part of the mechanism is allergic sensitization. Late onset asthma, that's T2 high, and that's caused by a Staph aureus enterotoxin. Aspirin exacerbated asthma, which we all used to say, oh yes, don't give aspirin to asthmatics because they get worse. Well, 1 or 2 of them do. That's due to genetic changes in arachidonic acid metabolism that's associated with T helper changes. But we've also got T2 low. That's non-atopic, and that has some very specific cellular and cytokine profiles that go with it. The smoking related, which is associated with oxidative stress, and obesity related, which is different again. Now, these again are made up diagnoses. Remember here, what we're doing is we're labeling them as atopic, and then we're looking for a molecular mechanism. And from all I've told you so far, I hope you realize that's a really stupid way to do things, because once again, it's becoming Dan Martin syndrome, Grocott syndrome, Montgomery syndrome, and then we try and find some markers of it, as opposed to going the other way around and saying, can we look at the biology and then define the disease state from that, which is the obvious and logical way to behave. So we're still seeking differences in profiles, by diseases we're defining as humans. And it's going to be just as flawed as it's always been since the days of diagnosing rheumatoid arthritis. Now, in our world of sepsis, or as I like to refer to it this way, I'm not alone probably in this audience in never really having believed in it. And it's easy to see why, because this was a fabricated myth that was invented years and years ago. And to my mind, it sits in there with unicorns and trolls. Because it first was diagnosed in 1989 from a post hoc analysis of 191 control patients from a steroid treatment trial. And all they wanted to do was try to say, could we find a way of picking out people who are at risk of getting worse? So it was really just saying, is there something we can look at that says this person might get worse? But to that got attached a name, and they called it Sepsis Syndrome. And suddenly, as opposed to defining a cluster of symptoms and that led to decline and just giving it a name that we could use, Victorian gentlemen in top hats, we suddenly said there was a thing that actually existed. We then said, well, actually, some of those features of the infection are found in non-infective cases. So that got a name as well. And that was Systemic Inflammatory Response Syndrome, or SIRS, which wasn't just used as a label. Suddenly it became a real thing. And then we put the two things together. There was another consensus conference that then said that you had to have SIRS features to define infection. And the criteria for SIRS were based on only 519 patients, all of whom were already in an intensive care unit. So in no way representative at all. So now we've invented something called sepsis syndrome, which doesn't exist. We've invented something called SIRS, which doesn't exist as a biological entity. And now we start trying to find treatments for it. And you can tell that this has been made up because it was redefined again in 2001. Both SIRS and sepsis were redefined. Severe sepsis was redefined. And then we redefined it again in 2016. So these are not real diseases. They're made up diseases. And it's stupid to think that you can go after a treatment for a unicorn. And just to quickly through, you can see why different organisms and different sites have totally different impacts. Having ascending cholangitis from E. coli is totally different from having a boil on your bum, which is totally different, let's say, from pneumococcal pneumonia, but we would call them all sepsis. The diagnostic features change. My sister next door had a temperature of 39.4 last night, and it's 36.8 this morning. The significance of each element differs. A high bilirubin is very different if it's a feature of generally being unwell from if you've got an infected obstructed biliary tree. We know that many features are not exclusive to infection. If you've had a head injury and a low GCS and aspirate, you could be defined as having sepsis because you've got multi-organ symptoms, including a low GCS. Sometimes infection's never confirmed. Well, does that actually mean it was ever there in the first place? Pediatric features are totally different. White cell count is useless in many cases because it could be high, low, or in the middle. And biomarkers of infection, sensitivity and specificity. So we're as guilty as anybody in the intensive care world, the world of some of you in this audience, for this sort of behaviour too. Sometimes, though, we recognise an illness and we do say, well, actually, we really can recognise it, but we just don't know what to do. Now, sometimes, of course, as I say, in the cases of, let's say, an appendicitis, you do recognise it's bleeding obvious and you know exactly what you should do, which is take the appendix out. But very often we can recognise the disease state, we think quite accurately, but we just don't know what to do. So let's look at this. Murderers are associated with fences, junk food, and knives and guns. Some of those associations are merely bystander associations. People in poverty will eat more junk food, and people who live in poverty tend to be involved in more crime than people outside. That's not to say everybody is like that at all, but there's an association. Fences are associated with murderers in a causative, well, in a direct relationship, because if you're caught for murder, you tend to be put behind a fence. But it doesn't mean that it caused you to be a murderer any more than the fact you used a knife or gun to commit the murder turned you into a murderer in the first place. So we don't actually know when we look at association what's just a bystander effect, what's a result of the state we're in, and what's a causative association, which makes studies like this, gene expression profiling blood for septic shock, bloody difficult to interpret, because the panel on the left, some things are going up, which is green, some things are going down, which is red, some things aren't changing, which is black. And in that melange, some things will be the thing that's causing you to be septic, some things will be a bystander effect, and some things will be as a result of being septic. And we have made this mistake throughout my medical career by getting activated protein C or monoclonal antibodies to TNF, for instance, because we take a punt on what might work. And that is buck stupid. Because if you do find an association that is causative and you target it with a treatment, the patient may well get better, which would be a very good thing. But if you target the thing that's an epiphenomenon, it either won't work or you will actually cause harm. And if the thing that you actually were targeting turned out to be a beneficial adaptive response from the patient, by blocking it, you're going to kill them off. And we have made these mistakes throughout my life in medicine, targeting things without actually knowing what the outcome will be. And often trials are stopped, not just for futility, but because of harm. Fortunately, though, times have changed. So we are no longer dealing with gentlemen in top hats defining disease, but we're able to apply appropriate biological methodologies to try to define disease states. Because we now have a sequence of the 6 billion bases of the human genome. And we know now that those 20,000 genes, when I was at medical school, it was one gene, one protein. There are roughly 20,000 genes. We now know that from post-translational modification, from splicing, and from selective expression, that there are potentially billions of proteins that make up the human proteome. But we can now find those billions of proteins and determine what they are, just as we can define the more than 40,000 bacterial species that make up our gut microbiome. So omics, the ability to examine metabolites, the metabolome, the genome, the proteome, the gut microbiome, all becomes possible. So now we might be able to start going back to front, look at those data, define clusters based on biochemistry and biology, and then define diseases based upon their molecular causation, not by what a gentleman in a top hat once said. I'm not going to give you a long talk about how you do this, but one can do this with genomics. So this is an application here of artificial intelligence with what's known as combinatorial genetics, where you can take the entire human genome sequence, look at all of the genetic variants, and then with a supercomputer can do up to 10 to the 50 combinatorial comparisons. And by using artificial intelligence, learning what those patterns look like and referring them through sequential samples from a Human Genome Project, but also from databases held in the UK, you can start finding these sorts of clustering patterns. On the left, they're rheumatoid arthritis, which we've spoken of, ALS in the middle, and asthma associated with COPD on the right. And you can see there are very, very distinct clusters of interacting genes in these sorts of people. And you can, in fact, start teasing them out by looking for differences in characteristics in rheumatoid arthritis. And some of those clusters then separate out completely from the others. So you can actually say, well, actually, this is a distinct disease in its own right. So this is combinatorial genetics. You can look at the expressome in cells. You can look at the RNA that is being encoded from the gene or transcribed. And by doing machine learning, you can start clustering that transcriptome and attach it to diseases. So there's one sort of cloud there on the left where you can see distinct expressomes clustering from blood for leukemia, heart failure, hypertension, diabetes, etc. But you also start seeing some of those expressomes are remarkably close. There's actually some overlap, as it turns out, between the expressome for ulcerative colitis and Parkinson's, just as there is with rheumatoid arthritis. And there's an overlap indeed with some genes with glioblastoma and type 2 diabetes. So some of these things, and indeed, of course, very well recognized now, a gene clustering overlap with asthma and multiple sclerosis, suggesting some common causative mechanisms. So you can do it with your expressomes. You can apply machine learning and produce these hill and valley plots, which I think are much easier to interpret to a human eye. We have the idea there are boundaries and these valleys separated by mountain ranges, where you can then start saying, well, these people are distinctly different from those people. And you can then start doing it agnostically and say, well, actually, that group seems to sit in something we currently would call, let's say, asthma. And you can produce bubble plots of this sort as well, different ways of just basically representing the differences in expression profiling. You can do other approaches. This is the interactome. So essentially, you draw down the proteins inside a cell and see which cells are which proteins are interacting with other ones. So in other words, which ones are stuck to other ones as ligand and receptor, for instance. And if you start doing that, you can identify all of those physical interactions. And for each of those little nodes, which is a protein, you might be able to see on the screen, there are little thin grey lines which connect those dots if there's an interaction between those proteins. Then you can start labeling the nodes, as it were, as ones that are associated with a particular disease. And if you join the dots up to what's known as a module, if it's one disease pattern. So you can see clusters of genes that seem to sit there with multiple sclerosis or rheumatoid arthritis, and we've commented on that overlap a bit, and some of the peroxisomal disorders. But you can also see how those genes interact and relate, or the expression proteins rather, interact with others. So you can start getting a handle on the mechanism of disease. So now we might have the chance to actually start defining diseases and understanding what causes them so that we can develop targeted treatments rather than giving, let's say, everyone with asthma a salbutamol inhaler and thinking that we can benefit them. And we're seeing this sort of difference now. So AstraZeneca have a new drug, which I think will be licensed in September or so, which can put people who are T2 high into complete remission from asthma. So isn't that extraordinary? By understanding the biology, which in this case is targeting eosinophils, you're able to prevent anybody getting an exacerbation of asthma, because actually we've understood what that disease is, and it isn't the same as other asthma. So, to recap where I'm at the moment, the first thing is I've demonstrated that I know nothing, and I would like to put it to you that in general, most of us know nothing. We've been living in cloud cuckoo land for years and years and years, and it's now becoming clear to us that we need to rethink how we define diseases and consequently how we set about trying to treat them. But the other thing is that I've now learned that the things I certainly knew for sure were generally wrong. And if I go back, some of you are as old as me. Mike is a bit younger, so is Dan. Monty is similar age to me. But he'll remember the days when we had a patient on the intensive care unit where we took the Frank Starling curve and we just said, keep fluid challenging them until there's no change in stroke volume. Get them to the top of their Starling curve. Well, that was a very good way of killing people by fluid overloading them. I was absolutely fantastically good. I reckon I was the best in the world at managing to recruit lungs and give people really whacking size tidal volumes. We patted ourselves on the back when we got people 1,200 mil tidal volumes. Look, we really expanded that lung. We were blowing all that CO2 off. How many people I killed in doing that, I don't know, but it will be a very large number, because in those days we thought that was the right thing to do, and we were confident that it was the right thing to do. And there are other things that we now know are nonsense. You know, the idea that you give everyone on ITU a lactulose to keep them pooing. All that does is mean you have to change the sheets a lot. But that brings us back to a lot of other things we knew for sure. So this is an ITU graph of patients who are transfused or not transfused in ITU, and you'll see that hemoglobins, as we all know, drop steadily over time. And in the old days, we used to jack red cells in because clearly part of ITU was to get people back to their normal homeostatic state. So let's give them that hemoglobin of 120 or 130. And that's what we used to do. But of course, it's the same as the guns, fences and hamburgers argument. Is that fall in hemoglobin just a bystander effect of irrelevance? Is it a consequence of being ill, which doesn't cause any harm? Is it a causative factor in people being ill, in which case we top it up? Or is it actually a benefit to the fall in hemoglobin, and by giving red cells, we kill people off? Well, of course, we've since learned that we probably were causing harm, and none of us now start jacking in red cells randomly to people because their hemoglobin is low, and most of us stick to this transfusion trigger of 70, because across most disease states, it's quite clear that for whatever reason, having a lower hemoglobin is either beneficial or giving them the red cells directly causes harm. Either way, you're worse off if you get the red cells. And I bring this back to our understanding of weight loss in intensive care, because I wanted to bring this back to home to Mike and Dan and Monty, who led the Xtreme Everest work, because this is really what the Xtreme Everest work in part was about. This is a muscle biopsy taken by Zudin Puthacheri, who is an absolutely fantastic academic intensivist, to whose PhD these data are attributed. So this is a muscle biopsy of a patient on day one, a 35-year-old, in fact, with malaria and multi-organ failure. Lovely, pink, plump, smooth muscle cells. And on exactly the same scale, this is what the muscle looks like at day 7. The cells are ragged, the interstitial spaces are full of edema, there's an inflammatory cellular infiltrate. Those cells are no longer staining homogeneously pink, they're staining for protein in a very patchy way. This is clearly a very diseased piece of muscle if I do that again for you. And of course, we all went, well, this is bad. We have to be treating that. We have to stop that wasting. It's clearly a really bad thing. Let me remind you, we don't know if it's gun, hamburger, or fence. Here's the muscle loss of volume, down by around 20% in rectus femoris cross-sectional area by ultrasound. But actually, if you look at protein content, it's down by around 50% in those muscle cells by day 10 in a critically ill patient. So here is Mike on the left, who set about trying to work out how this happened and why it happened, along with Monty on the right. That's not Monty on the extreme right. I hope you understand that isn't Monty when his first name was Sheila. That's Monty, the rugged mountain goat, standing there to the left. And what Mike showed at altitude was what we all know. You eat very well at altitude and you lose weight. And a lot of that is lean muscle mass. It doesn't matter if you're sitting around at base camp or if you're like Mike and you're bounding up to the summit with Dan. All of them lost a lot of weight. And we know now that this is a programmed process. So taking you through from the left top, the sarcolemma, there are some amino acids coming in, or there's some loading, that's you going down the gym lifting weights, or there's a hormone, insulin-regulated growth factor. And through a series of different signaling pathways, you reach this thing called mitochondrial targeted rapamycin, or mTOR. And if you switch those things on, you grow muscles. So in other words, if you eat your protein powder, go down the gym, and you sneak in a little bit of naughty IGF from your local drug dealer, you'll end up looking like Arnold Schwarzenegger. If, however, you become hypoxic, you switch on something called FOXO, which switches off those pathways, and you inactivate the ability to grow muscle. So when you go hypoxic, you switch off muscle growth, but you also switch on protein breakdown. So this isn't some sort of random badness that you would try to stop. This is a programmed process for which you'd expect there must be some biological benefit. And what Mike and I and Zudin was the first author on these papers, went on to show that muscle mass reduction in mountains was caused by metabolic processes through HIF. And we then went on to show that the muscle mass reduction on ITU is also metabolically driven. And we did that by doing the sorts of analyses which we spoke of earlier. And this one is called Markov Clustering, where you take a whole load of proteins from the muscle and you see what goes up, what goes down, what stays the same, and you color code them. And then you link them together with lines. Red means one thing goes up, the other one goes down. Green means that one goes up, the other one goes up. And the more tightly that association exists, the closer you put the dots together. So this is Markov clustering. And what we've shown here, if I take that little bit out, is that you've got mTOR and HIF, that very same pathway that we showed with hypoxia, is driving the muscle breakdown. So the system that we see on Everest is hijacked here in intensive care. And Mike and Dan and Monty's hypothesis originally was indeed a good one, that actually this was hypoxic. The heart wasn't pumping enough blood, you systemically were hypoxemic, that the microvasculature was knackered. It turns out we right in part, but in fact, this system is just being switched on by pro-inflammatory cytokines. And when you switch on HIF with cytokines, even in plenty of oxygen, it just activates the hypoxic muscle wasting. So we now understand that in intensive care, when you're critically ill, you have a programmed process to switch muscle growth off. So here we are, same sort of process, cytokines working by HIF, switches off muscle anabolism and switches on the catabolism through exactly the same pathways. You can learn a few more things as well. If you look at the bottom left, there's a bunch of things that interact that have got absolutely nothing at all to do with the muscle growth. And those things are lipids, monounsaturated fatty acids, phospholipids, and indeed PUFAs, showing that the amount of fat you put in makes no difference at all to muscle growth. None at all. You can pour in as much fat as you like and it will not protect you. And yet, what do? Most of the calories we give in enteral feed or TPN are in the form of fat, which isn't going to help preserve your muscle mass at all, even if you wanted to. In fact, when you do look at these biological pathways properly, you find that you're switching off the ability to burn fat. You're switching down CPT1, MCAD, this electron transport factor. These are all elements that are useful in beta oxidation of fat, and yet you're switching them off. What are we doing? We're giving lots of fat because, hey, calories must be good for you. When your body is saying, I don't want to eat, I don't want to burn fat, please don't give me any at all. And at the same time, we're trying to drive oxygen to muscle cells when actually you're stripping out the mitochondria that would burn them. This is crazy. We are doing things that are biologically not sensible, but we did them with the best will in the world. So if the body's trying to reduce its metabolic demand by reducing protein synthesis, getting rid of fat oxidation, getting rid of mitochondria, it doesn't seem very much of a good idea to give that fat. It doesn't make much sense to give lots of oxygen. So this was a good trial in the days, again, that we all part of this, the Schumacher hypothesis. Drive more oxygen, the body will burn more oxygen, and the body will do better. And David Watson, Michelle Hayes, and others did this study demonstrating not only futility, but harm, and that trial was stopped. We tried to keep muscles big because clearly muscle weighting is a bad thing on intensive care, isn't it? Nope, you do that one with growth hormone and it kills people, and the trial has to be stopped. And the same sort of applies when we try to feed aggressively with fat, because early full nutrition, if you really target it and you try to push, is associated in general with, at best, no benefit, and quite often with harm. Because again, what we've done is we've just measured something and we've decided it's a gun, not a hamburger. And indeed, there's good evidence now that starvation, and this is a different story, might actually be a good thing when you do the biology. Starvation produces ketogenesis. Ketogenesis provokes big changes in your biological functional immune system and might actually give you a very beneficial response to bacterial infection. So Granny was right, feed a cold, but starve a fever, because starving a fever is beneficial. So back to my youth again, my final words before I take some questions. It isn't just about the diagnosis. I still don't know what blood pressure to aim for in ITU. I think I should individualize it. I often try and crank it up to see what happens if the urine output's a bit low, but I don't know. Is there a universal target? Is there an individual one? And how would I define it? I don't know whether cardiac output monitoring helps. My strong suspicion that it doesn't. My strong suspicion is that if I put my finger on the patient to feel if they're warm, and if I see if there's urine coming and look at the lactate and feel their carotid pulse, that I'm getting more information than a cardiac output monitor. But I don't know. I don't know whether lactate is a good thing or a bad thing. It's a marker potentially of stuff happening in cells, but is the lactate something I need to get rid of? I suspect it probably isn't. Should I feed patients with multi-organ failure? Well, I don't know the answer to that question either. My strong suspicion is that if I was critically unwell and wanted to come to ITU, that I'd want you to starve me for the first 10 days. I can't prove that. I'm not advocating it to you. I'm just saying I don't know. How long should we give the antibiotics for? How do we monitor it? I don't know. How much oxygen should I give? Dan is trying to crack that one at the moment because we don't know. Let alone prognostication. We've all done this. You pull out on someone and then they send you a thank you card 4 months later on holiday in the Bahamas saying thank you for looking after them so well, when you were actually winning the Black Crow Award. So I do know nothing. And I still think I have pretty much no idea what I'm doing. This is not, by the way, to go onto my portfolio or into my litigation through the GMC, because we don't know what most diseases are. We don't get our own labels right a lot of the time. If you look at the data, we're misdiagnosing, and we don't know what we're meant to be targeting. And I think it's time, genuinely, for us to reappraise things and start again. We need to go back, redefine diseases by their biology, define how to treat them based on that biology, and we need to stop accepting the tenets we've all been given throughout our careers and accept that maybe we've been misinformed and maybe we've misinformed ourselves, and have a bit of humility and start again.
to say, brilliant, Hugh. I was just reflecting on the fact that I agree with almost everything you said. At times it could come over perhaps as a little bit nihilistic, but I think as one gets older and older working in intensive care, as you know, I've just very recently retired clinically, you still do get the impression that the intensively caring component of intensive care is enormously important. It's the thing where we possibly went adrift, was getting too clever with all the things that you described. So being a little bit nihilistic for a moment, I think you're nodding, intensively caring is important.
Yes, and my sister, who's behind me, won't mind me saying, he's had a terrible week with metastatic breast cancer and is transformed by love around her and by good palliative care. She is a different person today than she was yesterday. And we do have to make sure that we don't torture people. It's something I've spoken of before. If you look at the CIA handbooks of how to break people, we do every single element of the 12 things on intensive care with the best of intentions. We have forced nudity, we have captors omnipotence, we show death around them, we cause pain and indignity, we give altered sleep-wake cycles, we deprive people of regular nutrition. All of these things, if you wanted to break someone, you would do deliberately. And it is something, I know you agree with me on this, Monty, we have to start thinking, while we do the hard stuff, we do have to try to do it in a way that's humane. And I think we may have lost our way a little bit. And, we all hold the hands, we all talk to the relatives and the patient, but perhaps we ought to think a little bit more holistically.
So some comments or questions from the audience, and if anyone online would like to ask anything on Slido, please do. If anyone wants to get their hand up and ask a question or make a comment, please do. Bit of stunned silence here, Hugh, from all of that.
Hugh, we've talked about this before. We seem to, and you've described several examples in your talk, we seem to, we find a solution to a problem, like a dialysis machine for kidney failure or a ventilator, and we assume the solution is best if we give most of it, and then we progressively backtrack. Where's it going to stop? Are we going to end up doing almost nothing, or?
Well, apart from intensively caring, which is probably what it was there for in the first instance.
The transformative things in my medical career where I really look at something where I go, wow, that patient was totally transformed, have very often been the surgical things. They've been a heart transplant or a kidney transplant or something where you take the bad one away, chuck it in a bin and give them a new one. And that's not to say that ventilating a young woman with pneumococcal pneumonia doesn't save her life and restore her, but those things are not difficult and complex medicine. I think what we're getting into now is throwing the book at people who are progressively decaying with advanced age and multi-organ failure. And whether doing the least and seeing who comes out the other side of it is actually a slightly more humane but actually logical thing to do. In some cases, I'm not sure. There was that study, must be 10 years ago, Monty, you can probably quote it from the States, where people with a poor prognosis were triaged to palliation or intensive care, and survival was longer in the palliative care group than it was in the intensive care group. So, and again, I don't know the answers. I don't know how to choose those people, other than to try to base it on maybe to be gentle and then see who does well, gentle and compassionate rather than necessarily too aggressive. That's not to say one shouldn't throw the book at a young COVID patient or whatever, but we have to try and work out, I think, navigate some way through compassion and in some cases doing a little less.
Hugh, thank you very much indeed for a wonderful, challenging lecture. Our thoughts are with you and your family. Good luck with everything. Look forward to seeing you soon.
Thanks very much indeed, Monty, and thanks for having me.
Top Med Talk. Thanks for downloading Top Med Talk. Don't forget to subscribe via your podcatcher. Don't forget to check us out on social media. We're on Twitter, Facebook, LinkedIn, and YouTube. And also, don't forget, Top Med Talk is the broadcasting arm of EBPOM, Evidence-Based Perioperative Medicine. We'd love you to find out more about that. If you check out ebpom.org, you can find low prices on some of the conferences we're organising around the world. Many of them are virtual and don't even involve you leaving your own home. Check out ebpom.org now.